I'm Professor Sims, and in this video I'll discuss Mendel's experiments and heredity. This is the second in the series of 10 lessons held as part of my general genetics course. If you're a student currently enrolled in this course, please consult the syllabus and course Moodle site for assignments, quizzes, due dates, and other course information. The learning objectives for this unit include describing and calculating expected outcomes of monohybrid crosses, applying the sum product rules to calculate probabilities, explaining Mendel's law of segregation, independent assortment, calculating probabilities of genotypes and phenotypes from multiple crosses, explaining the effect of linkage and recombination on gamete genotypes, and identifying non-Mendelian inheritance patterns. Genetics is the study of heredity. Johann Gregor Mendel laid the foundation for genetics long before the discovery of chromosomes or genes, when meiosis was poorly understood. Mendel conducted meticulous quantitative analyses on a simple biological system with large sample sizes, revealing the fundamental principles of heredity. We now know that genes carried on the chromosome are the functional units of heredity responsible for replication, expression, and mutation. Mendel's postulates form the basis of classical genetics, although not all traits follow Mendelian patterns. His experiments provide an excellent starting point for understanding inheritance. For his experiments, because it naturally self-fertilizes and it produces true breeding plants. So this allows for him to avoid unexpected traits popping up in the offspring. The garden pea also has a short life cycle, so that allows for evaluation of multiple generations within a short period of time. And then large quantities of pea plants can be cultivated at the same time, so that reduces the role of chance affecting the results, because you have a large sample size. In his crosses, Mendel mated true breeding individuals with different traits, and this is known as hybridization. He manually transferred pollen from one plant to another plant in order to prevent self-fertilization, and then the offspring of these crosses were called the F1 generation. And then Mendel allowed the F1 generation to self-fertilize naturally, and that produced the F2 generation. And Mendel observed a 3-to-1 ratio of dominant to recessive traits in the F2 generation, and this demonstrated the independent transmission of trait.
Traits are characteristics or features that can be passed from one generation to another. Some examples include hair color, height, skin color, or gender. With dogs, it could be long tails versus short tails, ears or short ears, or maybe even the shape of the nose. Each of these factors can be passed genetically to another generation. There are different forms of the same trait. In this next video, notice the different shapes of the noses. These would be examples of alleles, because they are different forms of the same trait. Trait can also be dominant or recessive. Each individual has 2 alleles for each gene, one coming from each female and the other from the male. With dominant traits, an individual carries 2 dominant genes, or 1 dominant and 1 recessive gene. A dominant gene can be expressed even if only 1 copy of the gene is present. A recessive trait will be expressed if an individual has 2 recessive alleles. In this example, the purple color is dominant over the white color. In summary, a trait is a feature that can be passed from one generation to the next.
Genes on chromosomes determine physical characteristics, and peas, like any other plant or animal, are diploid organisms with 2 copies of each chromosome. These copies have the same gene order, and then during fertilization, haploid gametes unite to create a diploid zygote. We saw this in the last lesson. In diploid organisms, each gene can have 2 different versions called alleles. Alleles are found at the same location on homologous chromosomes, and then they have some variation that has arisen from mutation. So it's common to find more than 2 alleles for a gene in a natural population. The physical traits that are expressed by an organism are called its phenotype, while its underlying genetic makeup is called the genotype. And Mendel's experiments with hybridization showed the difference between the phenotype and the genotype.
So Mendel performed a lot of experiments on different characters, right? So let me take one example, the character height. So what he would do in one of his experiments is he would take a pure tall pea plant and then cross-fertilize it with a pure short pea plant and then see what offspring they would give. Now, I'm pretty sure you may be wondering, what do you mean by pure plants? What does this pure mean? Pure basically means if you were to self-pollinate them, so if you were to self-pollinate a tall plant, it should give all tall plants. If you self-pollinate a short plant, it should give all short plants. And what do you mean by self-pollination? Well, suppose this is one of the flowers of the tall plant. Then what I mean by self-pollination is you take the, what do you call this? Pollen grains, yeah. You take the pollen grains from the flower and put it back into the carpel of the same flower. Basically, you put it back into the female reproductive part of that same flower. And then it'll grow seeds. And then you sow those seeds. And then if you find that all the seeds grow into tall plants, then we will say this is a pure tall plant. This is the meaning of pure. And similarly, if you were to do the same thing for the short plant as well. So you take one of the flowers of that short plant and then you self-pollinate, meaning you take the pollen grains, put it back into the same flower, let it fertilize, and then you grow seeds and you sow them. And if you find all of them grow into short plants, not a single tall plant, but all of them grow into short plants, then we will call this a pure short plant. Okay, so Mendel prepared these pure breeds for his experiment, a pure tall plant and a pure short plant. Let me get rid of these now. And so for his experiment, you know what he does? He cross-pollinates them, meaning he takes the pollen grains from one of the plants and he puts it into the flower of the other. And you can take it from anyone to anyone. So for example, he might take the pollen grains of flower from the tall plant and then put it into the carpel or this part of the short plant. So let's say he cross-fertilizes them like this. And then this will start growing seeds. Then he sows that seeds to see what we will get after this cross-fertilization. So what do you think we will get? Well, let's get rid of the flowers and make some space. So what do you think we end up with? Well, I would expect to get some medium height plants, right? Because you have a short one, you have a tall one, you have a short one, so maybe they get mixed and you end up with all medium-sized plants. That's reasonable to think, right? But to Mendel's surprise, and to even my surprise when I learned this, we get all tall plants. What? That's right. Big shock. 100% of them end up being tall. This raises many questions. What happens to the short one? Does it have no effect at all? Why aren't we getting any medium-sized plants? Why aren't we getting any short plants? What is going on? Anyways, he addressed these plants as F1 generation plants. That's just the name that he gives them. And if you are wondering why he called them F1, the F stands for a Latin word filial. I hope I'm pronouncing that right. It means son or daughter. Okay. So that's why these plants are called F1 generation. First generation sons or daughters of these, you can say. But yeah, Mendel was shocked by this. So he didn't stop over here. He started asking questions. One of the main questions he was asking is, are these plants, these tall plants, the same as the parent tall plant? Are they same? They look the same because they have the same height, but are they really the same? Is this also a pure tall plant? That's the question he started asking. And to answer that question, you know what he did? He did another experiment. He didn't stop over here. What he does next is he takes one of the F1 generation plant, which is basically tall. He takes one of the F1 generation plant. This is F1 generation. Oops, let me use this white color. So this is F1 generation. And he self-pollinates it. Self-pollinates it to see what we get. So again, it will grow seeds, and then he sows that seeds. What do you think we'll end up with? Well, surprise, surprise, now some short plants are found. Another shocking thing to see, right? Because again, where did these short plants come now from? Certainly, right? And guess what? Mendel even counted them. So in this F2 generation, this is the second generation sons and daughters, or offsprings. When he counted them, you know what he got? He found that there were roughly 3 times as much tall as you had compared to short. So the tall to short ratio was 3:1. And if you're wondering, yes, he also cross-pollinated the F1 generation plants, and he gets exactly the same result, 3:1. And you know why this number is so important? Because it's not random. It turns out he gets the same answer for all the characteristics that he experiments with. I mean, for example, when he takes the color of the flower into consideration, again, he takes a pure purple-colored pea plant and crosses it with a pure white pea plant. In the first generation, you know what he gets? A similar result. This time he gets all purple-colored flower, no yellow, no white flowers at all. But then, when he takes one of these F1 generation purple flower and self-pollinates, guess what he gets? He now gets 3 times as much purple as white. White comes back. It's in minority, some small amount, 3:1 ratio, but it comes back with the same ratio, 3:1. So what's going on? Why didn't we see the white flower over here, but somehow it comes back in the next generation? What is going on? Before we think of what's really going on, one thing is very sure. Our initial assumption that traits get mixed and sent to their children is absolutely wrong, isn't it? I mean, if that was the case, we would have found some medium plants over here. Or over here, we would have found some pinkish colored flower, mix of purple and white. But we don't get that. So Mendel, from his experiments, conclusively proved that traits do not get mixed at all. That in itself was a huge discovery. But now he had to figure out what it all meant.
The reason why the F1 heterozygous offspring resemble one parent is due to the dominant and recessive alleles. So Mendel called dominant traits those that were inherited unchanged in hybridization, and then recessive traits were latent and then reappeared in subsequent generations. So Mendel proposed that plants possess 2 copies of each trait, and each parent contributes 1 copy to their offspring. Mendel's findings, based on the principles of probability, revealed the basics of heredity, because he was able to demonstrate that traits were transmitted as discrete units. They were independent of each other, and then by applying the product and some rules of probability, Mendel was able to predict the outcomes of the crosses. When true breeding plants with yellow and green pods were crossed, the hybrid offspring all had yellow pods. However, when the green pod allele reappeared, it was in later generations, indicating that there was a difference in the genotypes. The parent plants were homozygous for the traits that he studied, meaning that they had 2 identical alleles for the gene. When contrasting traits were cross-fertilized, the offspring were heterozygous, having different alleles for the genes. When talking about genes and alleles, we have a few conventions that you should know. We'll see the first letter of the dominant trait as an abbreviation for the gene. So if violet is the dominant trait for flower color in a pea plant, we use capital V to represent the flower color gene. Additionally, we use uppercase letters for dominant alleles and lowercase letters for recessive alleles. So homozygous dominant pea plant with violet flowers is represented as capital V, and then a homozygous recessive plant with white flowers is lowercase v, and then heterozygous plant with violet flowers is capital V, lowercase times V. Let's look at a monohybrid cross. So when two true breeding parents with different traits are crossed, it's called a monohybrid cross, and the resulting offspring are called monohybrids. Mendel conducted 7 monohybrid crosses, each involving traits that contrasted with each other, and his findings in the F1 and F2 generations showed that each parent contributed one of the 2 paired unit factors to their offspring, and all combinations of these unit factors were equally likely. To illustrate a monohybrid cross, let's consider true breeding pea plants with yellow and green seeds. The dominant seed color is yellow, so the parental genotypes are Y for yellow seeded plants and lowercase y for green seeded plants. We can use a Punnett square created by Reginald Punnett to predict the possible outcomes of a gamete cross and their expected frequencies. The Punnett square lists all possible combinations of parental alleles and shows the resulting diploid genotypes of the zygotes. From the Punnett square, genotypic and phenotypic ratios can be determined. For a monohybrid cross with true breeding parents, there's only one possible genotype, which is uppercase Y, lowercase y, resulting in all offspring having yellow seeds. If we do a self-cross of one of the heterozygous offspring, we can use a 2x2 Punnett square because each parent can donate one of the 2 different alleles. This results in 4 possible allele combinations. You have Y, lowercase y, capital Y, or lowercase y. And it's important to count both possibilities of obtaining the heterozygous genotype, a capital Y from the egg and a lowercase y from the sperm, or a lowercase y from the egg and a capital Y from the sperm, right? So that's where the difference is where they're getting them from. And despite their alleles coming from different parents, both combinations yield offspring that are genotypically and phenotypically identical. Since fertilization is random, each combination is equally likely, leading to a genetic ratio of 1 to 2 to 1, heterozygotes being the 2, right? So the ratio is 1 homozygote dominant, 2 heterozygote, and 1 homozygote recessive. And then Mendel confirmed these results by doing an F3 cross, which is self-crossing the F2 plants. And when the green-seeded plants were self-crossed, all the offspring had green seeds, confirming that green seeds have the homozygous genotype lowercase y. In the self-cross of the yellow-seeded plants, 1/3 of the plants had yellow seeds, while 2/3 exhibited a 3-to-1 ratio of yellow to green. So the true breeding plants had the homozygous genotype lowercase y, and the segregating plants had the heterozygous genotype. Self-fertilization of these plants produced the same results as the F1 self-fertilizing cross. Here's another way to look at it. So this is what is called a test cross. Mendel developed the test cross to determine if an organism is expressing a dominant trait because it's a heterozygote or is because it's a homozygote. So in a test cross, the dominant expressing organism is crossed with a homozygous recessive organism for the same trait. If the dominant expressing organism is a homozygote, all the F1 offspring will be heterozygotes expressing the dominant trait. Alternatively, if the dominant expressing organism is a heterozygote, the F1 offspring will have a 1-to-1 ratio of heterozygotes and recessive homozygotes. The test cross confirms Mendel's idea that unit factors segregate equally. Let's talk about alternatives to dominance and recessiveness. Mendel's pea plant experiment showed that genes come in pairs, with each parent contributing one allele. The dominant allele takes precedence, hiding the recessive one and not contributing to the phenotype. So recessive alleles can be carried without being expressed. And individuals with one dominant and one recessive allele are sometimes called carriers. Further studies in other plants and animals have revealed more complexity, but the basic principles of Mendelian genetics still hold true. One extension of Mendel's ideas is incomplete dominance. Sometimes the phenotype of heterozygotes appears to be intermediate between the parents. For example, in snapdragons, crossing a homozygous white-flowered parent with a homozygous red-flowered parent produces offspring with pink flowers, and this is known as incomplete dominance. In this case, the genotypic ratio would be 1 to 2 to 1, and the phenotypic ratio would also be 1 to 2 to 1. In codominance, you have both alleles simultaneously expressed in the heterozygote. So an example of this would be blood groups in humans. You have the M and N alleles, and they're expressed as M or N antigens on red blood cells. And then homozygotes express only one allele, while heterozygotes express both equally. Mendel suggested that only two alleles, one dominant and one recessive, exist for a given gene. But we now know that this is an oversimplification. Multiple alleles can exist at the population level, resulting in many combinations. And the most common phenotype or genotype among wild animals, this is designated as the wild type. Multiple alleles can exhibit dominance hierarchies. For example, in rabbits, there's 4 alleles for coat color, with the wild type allele expressing brown fur, the chinchilla allele expressing black-tipped white fur, and the Himalayan allele expressing black fur on the extremities, and the albino allele expressing white fur. So this is an example of a trait that has multiple alleles. And notice that there's not a huge difference in the ratios. It's just that these intermediate genotypes exist. And the result is not just brown or white fur. It's brown, white with black on the tips, white with black on the extremities, or just white. In sex-linked traits, the sex of an individual is determined by sex chromosomes. Humans have 22 pair of autosomes and then just 1 pair of sex chromosomes. Females have 2 X chromosomes, while males have 1 X and 1 Y chromosome. And then there is this phenomenon called hemizygous, meaning that you can have only 1 allele for X-linked characteristics. And then that's generally happening in males, whereas females, they have 2 X's. Genotypes and phenotypes of the offspring in an X-linked cross depend on the expression of the recessive trait by the male or female in the P1 generation. So eye color in fruit flies is one of the first X-linked traits studied. The wild type eye color is red, and red is dominant over white. If the male expresses the white-eyed phenotype and the female is homozygous red-eyed, then that means that all of the F1 generation will have red eyes. F1 females will be heterozygous. F1 males will all have white eyes. The discoveries made in fruit fly genetics can be applied to humans as well. X-linked recessive disorders such as red-green color blindness and hemophilia types A and B affect males more frequently than females. Females need to inherit recessive X-linked alleles from both parents to express the trait, while carrier females can pass on the trait to their male children or transmit the recessive allele to their female children, who then also become carriers. While some Y-linked recessive disorders exist, they're typically associated with male infertility, and because of that, they're not usually passed on to subsequent generations. Most of your sex-linked disorders are going to be on the X chromosome. Let's have a look at figure 1213, and this is giving an example of female carriers and heterozygous carriers and how things are inherited differently in males and females. A male is going to be XY, a female is XX. This orange color is affected, so let's say that this is hemophilia. Mom is a carrier. Dad doesn't have it at all. If mom passes on the X that has the dominant allele, so the dominant allele in this case would be not hemophilia, and she passes that on, then she'll have either a carrier or unaffected male. If she passes on the X-linked recessive allele, then she can also have a carrier or an affected male. And the dad, he can only pass on the dominant X or the Y, so he doesn't have the X-linked recessive allele at all. If any of the children have it, they're getting it from mom. I want to show you a video that goes into this process in just a little bit more detail.
Would you like to know one of the most underappreciated pieces of cytoplasm out there? Platelets. We take for granted the function of our platelets, which are fragments of cytoplasm that help stop us from bleeding. They help our blood clot when we get hurt. But there is a disorder called hemophilia that can affect those platelets because special clotting factor proteins that are needed to work with the platelets may not work correctly. So even a basic cut could be dangerous for people with hemophilia because they could bleed continuously. We have many treatments for the symptoms of hemophilia, and this has greatly improved outcomes with this disorder, although it wasn't always that way. Hemophilia is a sex-linked recessive trait, which means it's different from the basic Mendelian genetic problems. What do I mean by that? Well, we still use the terms dominant and recessive for alleles, but this time, because it's sex-linked, the alleles are on sex chromosomes. This is the case with sex-linked traits. So what is a sex chromosome? Well, recall that humans have 46 chromosomes. Chromosomes are made up of DNA and protein, and they contain your genes. Well, 2 of your 46 chromosomes, they are called the sex chromosomes. In a karyotype, it is usually the last 2 chromosomes that are the sex chromosomes. The sex chromosomes are called X and Y chromosomes, but it has nothing to do with the shape of the chromosome. That's always kind of confusing, but please don't think that Y chromosomes are shaped like a Y and X chromosomes are shaped like an X. This used to always confuse me. It has nothing to do with their name. The reason they got their name is actually kind of interesting, so to the Google for that. Everyone has an X chromosome, but if you have another X chromosome, meaning you have 2 X chromosomes, you are female. And if you have a Y chromosome, meaning you have an X and a Y chromosome, you are male. There are also genetic disorders where you can have extra copies of sex chromosomes, but we will not go into that in this clip. Sex-linked traits are traits that are specifically on the sex chromosomes. Most sex-linked traits tend to be on the X chromosome because it is larger than the Y chromosome and it contains more genes in the Y chromosome. The disorder hemophilia is like this. Now we will use the letter capital H to represent an allele for having normal blood clotting function and a letter lowercase h to represent an allele for having hemophilia. Hemophilia is a sex-linked recessive disorder, which is why it's being represented by a lowercase letter h. Only it must be placed on the sex chromosome as a superscript, like an exponent. A woman that does not have hemophilia could have the genotype X big H, X big H, or X big H, X little h. Because as long as she's got at least one dominant allele, that dominating allele will be what shows. So no hemophilia, since again, hemophilia is a recessive sex-linked disorder. The only way for her to have hemophilia would be the genotype X little h, because only when there is no dominant present will that recessive show up, at least in this type of trait. Now, for a male to not have hemophilia, his genotype would have to be X big H, Y. Notice how I did not put anything on the Y chromosome. Again, most sex-linked traits are on the X chromosome. If he has the genotype X little h, Y, then he has hemophilia. He doesn't have 2 X chromosomes, so he cannot be a carrier. So let's say that 2 people that do not have hemophilia have children. However, let's say that the woman is a carrier. That means she is heterozygous. How do you do a sex-linked Punnett square cross for this kind of trait? Step 1. Identify the genotypes of the parents. So the mother was a carrier. She is X big H, X little h. She does not have hemophilia because of that dominant allele present, but she is a carrier. The male, if he does not have hemophilia, well, he must be X big H, Y. There's no other option for him. Step 2. Place one parent on the top, outside of the square like this, and place the other parent on the left, outside of the square like this. Step 3, cross them. For formatting purposes, place X chromosomes before Y, and you also write any sex chromosomes with dominant letters first. The results you get in the squares would be the offspring, the babies. The genotype ratio could be written out like this. The phenotype ratio, remember that these are the observable traits, can be written out that there is a 75% chance that the child will be born without hemophilia and a 25% chance that a child would have hemophilia for this boy here. You could also make this a ratio, so 3 to 1. Notice that in this type of example of a sex-linked recessive disorder, males are more likely to inherit this disorder because there is only one X chromosome present. This is true for many other sex-linked recessive disorders, such as colorblindness.
Now let's talk about the concept of lethality. Many genes in our genome are crucial for survival, but occasionally a non-functional allele for an essential gene can arise through mutation and be passed on if individuals with this allele also carry a functional copy. The functional allele ensures survival and is considered dominant over the non-functional one. However, imagine you have two heterozygous parents with a wild-type non-functional mutant genotype, an essential gene. In a quarter of their offspring, we would expect individuals with a homozygous recessive genotype for the non-functional allele. Since the gene is essential, those offspring might fail to develop, and then they would probably perish in utero, or they might die later in life depending on when this gene is required. So this inheritance pattern where the allele is lethal in the homozygous form, but may show non-lethal phenotypes in the heterozygotes, that's called recessive lethal. When crossing heterozygous individuals with a recessive lethal allele causing death before birth, only the wild-type heterozygotes and homozygotes will be observed. So the genetic ratio would be 2-to-1. In other cases, the recessive allele may exhibit a dominant non-lethal phenotype in the heterozygotes. An example of this is the recessive lethal curly allele in fruit flies that affects wing shape in the heterozygote form, but it's lethal in the homozygote. Having one copy of the wild-type allele isn't always enough for normal functioning or for survival. In the dominant and lethal inheritance pattern, the allele is lethal both in the homozygote and heterozygote forms. However, for the allele to persist, the lethality must occur after reproductive age. Individuals with dominant lethal alleles do not survive, even in the heterozygote form. So dominant lethal alleles are rare because, as you might expect, the allele only lasts for one generation and it's usually not passed on. But dominant lethal alleles may not be expressed until adulthood, and once the individual reaches reproductive age, they might unknowingly pass on the allele, resulting in delayed death in both generations. Example of this is Huntington's disease, where the nervous system gradually degenerates. Heterozygotes for the dominant Huntington allele will inevitably develop the fatal disease, which may only manifest around age 40, by which time they may have already passed the gene on to about half of their offspring. Mendel had 4 postulates, often called laws, from his pea plant experiments, summarizing the basis of dominant and recessive inheritance in diploid organisms. However, as you've learned, there are more complex patterns of inheritance beyond the classic 3-to-1 phenotypic ratios. Yet, these laws provide a foundation for classical genetics, so let's have a look. The first one is the existence of paired unit factors, or genes, that are transmitted faithfully throughout gametatogenesis and fertilization. Contrary to the prevailing belief of parental traits blending in offspring, Mendel's experiments showed that heredity factors are discrete units. The second one, the law of dominance, states that in a heterozygote, one trait will conceal another trait for the same characteristic. Only the dominant allele is expressed, while the recessive allele remains latent but can be passed on. Offspring need 2 copies of the recessive allele to express the recessive trait. However, researchers have discovered that the law of dominance is not always applicable. Mendel proposed the law of segregation after observing true breeding pea plants with contrasting traits produced F1 generations with the dominant trait and F2 generations with a 3-to-1 ratio of dominant to recessive, ensuring equal chance of inheriting either factor. The F2 generation can have 3 possible genotypes: homozygous dominant, heterozygous, or homozygous recessive. Since heterozygotes can arise through different pathways, they are phenotypically identical to homozygous dominant individuals, supporting the 3-to-1 ratio. The physical basis for this segregation lies in the first division of meiosis, where homologous chromosomes segregate into the daughter nuclei. So let's have another look at independent assortment. According to Mendel's law of independent assortment, genes do not influence each other when it comes to sorting of alleles into gametes. Every possible combination of alleles for each gene has an equal chance of occurring. So we can understand and independent assortment through a dihybrid cross, which involves two true breeding parents with different traits for two characteristics. For example, look at pea plants with green wrinkled seeds and yellow round seeds. Since both parents are homozygous, the law of segregation tells us that green wrinkled plants' gametes will be lowercase y, lowercase r, while yellow round plants' gametes will be uppercase y, uppercase r. Therefore, all offspring in the F1 generation will be uppercase y, uppercase r, lowercase r. So they're all going to be heterozygous for both traits. Now, in the F2 generation, the law of segregation states that each gamete must receive either a capital R allele or a lowercase r allele, along with either an uppercase y allele or a lowercase y allele. Sorting of the lowercase r allele is equally likely to be paired with an uppercase y or a lowercase y. This leads to 4 equally likely gametes. When we self-cross the uppercase y, uppercase r, lowercase r heterozygote, we get uppercase R, uppercase Y, uppercase R, okay? Uppercase Y, lowercase y, uppercase R, and lowercase y. And by arranging the gametes in a Punnett square, we obtain 16 equally likely genotypic combinations, and from these genotypes we can infer a phenotypic ratio of 9 round and yellow to 3 round and green to 3 wrinkled and yellow to 1 wrinkled and green. And these ratios are what we would expect from a large enough sample size. Due to independent assortment and dominance, we can collapse the 9:3 to 3:1 dihybrid phenotypic ratio into 3:1 ratios, similar to what you would see for a monohybrid cross following a dominant and recessive pattern. By focusing only on seed texture or seed color, we would anticipate that 3/4 of the F2 generation will exhibit the dominant trait, while 1/4 are independent events, allowing us to apply the product rule. The physical basis for the law of independent assortment lies in meiosis 1, where homologous pairs randomly line up in orientations, and each gamete can contain any combination of parental and maternal chromosomes, the genes carried on them, to this random orientation. When dealing with more than 2 genes, the Punnett square method becomes kind of impractical, so we can use what's called a forked line and probability method. The forked line method involves creating rows for each gene and segregating the alleles along forked lines based on the probabilities for individual monohybrid crosses. So multiplying the values along each path gives us the probabilities for the F2 offspring. This is a representation of the product rule. So for trihybrid crosses, the F2 phenotypic ratio is 27, 9, 9, 9, 3, 3, 3, 1. Let's have a look at a figure. Figure 12.17 shows the fork line method used to analyze a trihybrid cross. So here, the probability for color in the F2 generation occupies the top row, right? So we have yellow, we have green. The probability for shape is in the second row. So we have 3 round, 1 wrinkled, right? And the probability for height is the third row. So tall and dwarf, 3:1. The probability for each possible combination of traits is calculated by multiplying the probability for each individual trait. So the probability of the F2 offspring having yellow, round, and tall traits is going to be 3 times 3 times 3, which is 27. And the probability of having green, wrinkled, dwarf plants is going to be 1 times 1 times 1, which is 1. And that's how you end up with the 27, 9, 9, 9, 3, 3, 3, 1 ratio. The probability method provides the expected proportions of offspring without the visual aid of a diagram. So it utilizes the product rule and considers the alleles for each gene separately. So in a tetrahybrid cross between heterozygotes for all 4 genes, the probability of offspring being homozygous recessive for all 4 alleles is 1/4 times 1/4 times 1/4 times 1/4, which is 1 out of 256. And then the probability of offspring having the dominant phenotype for all 4 loci would be 3/4 times 3/4 times 3/4 times 3/4, which is 81 out of 256. Here's a table that can help you when you're looking at things that have more than 2 crosses. These are the methods and rules that help us to predict the genotypes and phenotypes of offsprings that are in a multi-hybrid cross and follow the principles of independent assortment and dominant recessive patterns. So this is a good framework here for genetics calculations. You have number of F1 gametes, number of different F2 genotypes, given dominant and recessive inheritance, the number of different F2 phenotypes, and then here are the number of heterozygous gene pairs. Some combinations of alleles violate Mendel's law of independent assortment, which states that genes are inherited independently of each other, right? So we now understand that genes located on the same chromosome can be linked and tend to be inherited together. However, the process of recombination or crossover allows genes on the same chromosome to behave independently. So recombination occurs during meiosis, and segments of homologous chromosomes exchange genetic material, right? So when genes are physically close on the same chromosome, they're considered linked, and their alleles are more likely to be transmitted together. So if genes for plant height and flower color are right next to each other on a chromosome, the tall and red alleles might be inherited together, and then the short and yellow alleles are inherited separately. And this linkage affects the outcomes of dihybrid crosses because it deviates from the classical 9:3:3:1 ratio predicted by Mendel. As the distance between the genes increases, the likelihood of crossovers between them also increases, and this makes them behave more like genes on separate chromosomes. Geneticists use the proportion of recombinant gametes to determine the distance between genes and construct gene maps. So this is figure 1218, and this is just giving, again, another demonstration of what crossing over looks like. So you can imagine if this crossing over and recombination is happening and you have genes that are very, very close to each other on the chromosome, that it's more likely that those 2 genes will be transmitted together rather than separately. And in reality, single observable characteristics are often controlled by multiple genes working together. Genes might act sequentially, they might complement each other, they might oppose each other to influence phenotypes. An example of this is epistasis. Epistasis is a form of gene interaction where one gene masks or interferes with the expression of another. It occurs when alleles at one locus are dependent on the function of a preceding or on a following gene in a pathway. So, for example, the presence of a separate gene is necessary for pigment production in mice, and the lack of this gene results in albinism, regardless of the allele, the coat color gene.
The wild-type color agouti is dominant to the solid color for black. That is why both agouti and black-colored babies are born. However, the gene C also affects the phenotype because it affects the pigment production. If the gene is recessive homozygous, fur coloration pigments are not produced. This results in the birth of white baby mice. I will further explain this using this Punnett square.
In here, 2 parents are of agouti color, just like you and your wife. They are considered as heterozygous for both genes. These are the gametes made by these parents. This Punnett square shows possible genotypes and phenotype of the offspring. The mice with at least one dominant A and C alleles for the relevant genes are agouti, and the mice with recessive homozygous A and at least one dominant C allele in the gene are black.
Therefore, the genotypes with recessive homozygous C gene will produce albino mice regardless of the gene A. This is because the homozygous recessive C gene inhibits pigment production. According to that, a cross between heterozygotes for both genes would generate offspring with this phenotypic ratio. This phenomenon is known as recessive epistasis.
Recessive epistasis is a polygenic interaction where a recessive homozygous gene controls the phenotype of another gene responsible for a trait. In albinism, the gene C is epistatic to the gene A. That means The gene C masks the effect of the gene A.
That concludes Lesson 2's material. I want to thank you guys for watching. Don't forget to do the reading. Check the description for more videos related to these topics and leave your questions for me in the comments below.